Welcome back, students. Let's deal with food hazards in overview. In the previous classes, we had come across the different hazards. What are the hazards like physical, chemical as well as biological hazards? We had discussed the various techniques which we can use to detect the hazards. And right now, we are at a stage at which we are trying to detect the microbiological hazards. In the last session, we had discussed the microbiological hazard detection using culture-based methods as well as the immunology-based methods. Right now, today we're going to discuss the polymerase chain reaction or the PCR-based method for the detection of food pathogens. Using the PCR technique, we can detect even a single copy of a target DNA sequence. A target DNA can be amplified to about 1 million fold in a short time of few hours using the PCR technique. And it is the most sensitive technique for the detection of Salmonella, Staphylococcus aureus, Bacillus cereus, etc. And another variant of the PCR called the multiplex PCR will help us detect different strains or different bacterial species using different primers. That is, in a single tube itself, you will take different primers and they will help us to amplify the DNA regions coding for specific genes of each bacterial strain targeted. So let's first go into the basics of PCR. PCR was developed in 1983 by Carey Mullis, and it is now a common and often indispensable technique used in medical as well as biological research labs for a variety of applications. The same technique can also be used in the food microbiology labs also. And the vast majority of the PCR methods use thermal cycling, that is, alternatively heating and cooling the PCR sample to a definite series of temperature steps. Now, the particular reaction of PCR, it requires the DNA fragment to be amplified. Now, for example, the genomic DNA or cDNA. In the case of food samples, we would get a sample which we will extract the DNA from the food sample. Then, 2 oligonucleotide primers specific to the DNA sequence of the desired fragment, a purified DNA polymerase, DNTPs as well as a buffer solution. Now let me just go into the details of this. Now, the DNA containing fragment to be amplified is, in the case of food samples here, it would be the DNA which is being obtained from the food samples. Then these are the primers. What are the primers? If you have to detect here E. coli, the primers are sequences which will help us to identify the presence of E. coli in the sample. They are short segments of DNA which will be tagging a gene specific of E. coli. Now, if you're trying to detect Salmonella typhi or Staphylococcus aureus, the primers will be specific for that particular organisms. Then, what is DNA polymerase? DNA polymerase is an enzyme which helps in the DNA synthesis. And DNTPs, they will serve as the building blocks for the DNA synthesis. And this, of course, is a buffer solution which will help us to maintain the particular pH during the DNA synthesis happens. And you also need a process called thermal cycling. Thermal cycling is a series of steps which will help us to denature the DNA. Okay, so at the first step is you take a DNA and it has to be denatured. You know, DNA is being found to be double-stranded. So to make it single-stranded, you give a denaturation step. And at the second step, what happens? You reduce the temperature to 60 degrees centigrade, and the primers, these are the single-stranded primers which will attach onto the single-stranded DNA, and further extension happens for a specified time. So every time what happens is, you can see that the denaturation, primer annealing and extension step will continue for different cycles. That is, you take a DNA, you will first denature it and then it will have a process of annealing, then it will have a process of extension, and this step continues almost 25 to 35 times, making multiple copies of a particular gene which you need to identify. So that is the reason why we call it as a polymerase chain reaction. You are using an enzyme called polymerase here, and this is being carried out for different cycles. So it's a chain reaction, that is denaturation, primer annealing step, and extension. Like that, the three steps are being followed one after the other to result in the formation of a huge amount of DNA, which will a huge amount of DNA of a small gene, not the entire DNA sequence, but a small bit of the DNA which helps us to identify the presence of the pathogen is being multiplied. PCR in fact is named as a photocopying machine of DNA. So if you want to identify an E. coli in your food sample, what you do is you will take a primer which is specific to E. coli, and the DNA from your food sample is being taken, and then you will put a primer which is specific for E. coli, and then if E. coli's specific DNA is being present in that, what happens? A copy of this E. coli DNA, which is specific to the primer you are given, will be amplified. That is, a part of the E. coli DNA will be amplified. Or many copies of that will be made during the PCR reaction, so that it becomes detectable for you within a period of about 2 hours or maximum 3 hours, depending upon those. Some PCR machines even give you the result in 1.5 hours. So the reaction time depends upon the machine that you use, as well as the primer which you have given, as well as the reaction mixture. So let's go and see how this happens. So repeated thermal cycling initiates new rounds of DNA replication that can use the products of the previous round as template, and thus exponentially amplifying the target DNA fragment. That means that every time a cycle is being completed, the products of that will be used as a template for the next cycle. So at each cycle, what happens? You are getting more amount of DNA after each cycle. Now let's look into the process, how it works. If this is a double-stranded DNA, okay, this is the fragment which has to be amplified. I told you not the whole DNA gets amplified, only a small segment of the DNA gets amplified. So if this is a gene, if this is a whole E. coli genome, which has been, if considering that E. coli is being present in the food sample, only small segment of this will get amplified. It has to be amplified. So what happens? When you give a denaturation temperature, the whole DNA gets separated into a single strand of DNA, as you can see in this figure. After that, annealing of the primer happens. The annealing happens at around 60 degrees centigrade. Okay, and what happens in the next step? Let's have a look. What happens? The DNA polymerase comes and binds, and the DNA synthesis happens. Okay. Now in the next step, what happens? Again, the denaturation will happen. The primers will come and bind, and DNA synthesis will happen further on. Again, denaturation, annealing, extension, denaturation, annealing, as well as extension. So with each repeated thermal cycle of this thing, you can see that the amount of the target double-stranded DNA doubles. So this was a strand of double-stranded DNA which we had to produce, and that with each cycle, what happens? The double-stranded DNA molecule will be found to be increased. So this is a machine which was used for the PCR. Of course, now you have different other techniques, other modern PCRs which have been available. This was the old model which was available, and usually this was mainly possible. The PCR technique was possible because an enzyme called the Taq DNA polymerase was being isolated, and it lacks the proofreading activity, and this was an enzyme which was used for the synthesis of DNA in the PCR technique. And now we can also use another enzyme called the PFU polymerase, which is extremely heat stable, and it is from the Pyrococcus furiosus that has also been available. So you have got two enzymes which can be used for the PCR. One is Taq DNA polymerase from Thermus aquaticus, and the second one is the PFU polymerase from the Pyrococcus furiosus. I hope the PCR technique is clear to you. We will be discussing the next techniques in the coming sessions. Thank you for now.